Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic, that is your introduction of aldose reducing hexose sugar. Okay, so here aldose means that the aldehyde group is present. Aldehyde group is present. Okay, reducing sugar are those sugar in which free aldehyde group or keto group present. So here aldose reducing sugar, then it means that free aldehyde group is present, and hexose means that 6 carbon atom is present, and the glucose is the free that glucose is known as that aldose reducing hexose sugar. Okay, so we have to discuss about the basic of that aldose reducing hexose sugar, that is your glucose. So let's start the lecture. Introduction. We all know that carbohydrates are the essential biomolecules. Okay, biomolecule means that this carbohydrates is the organic compound, and it is made up of carbon, oxygen, and hydrogen. Okay, and this carbohydrate is the primary source of the energy. This is a primary source of energy. And these carbohydrates are essential for all type of the living organisms. And among the carbohydrate, this glucose is the most common type of the carbohydrates, and even this glucose is very important. Apart from that primary or most common, this glucose is very important. Okay, glucose is a monosaccharide, means that we cannot further hydrolyze it. We cannot further hydrolyze it. Okay, especially an aldose reducing hexose sugar means that here a free aldehyde groups are present. That's why we call it a monosaccharide aldose reducing hexose sugar, means that the free aldehyde group is present and 6 carbon atom is present. Okay, and it is the primary source of the energy. Okay, so this is the introduction about it. Carbohydrates are the essential biomolecule found in almost all living cells. Here, the living organism means that the plants, animals, and human beings. Plants, animals, and human beings all come in this category. And for all the living beings, this carbohydrates are considered as that primary source of the energy. And among the carbohydrate, we can say that the glucose is the most common type and important carbohydrates, which is your monosaccharide, means that we cannot hydrolyze it. It is the simplest sugar or smallest unit of carbohydrate. We can say that the smallest unit of that carbohydrate, and especially an aldose reducing hexose. Okay, so this is a introduction part. Now come to the next slide. Classification of carbohydrates. So Carbohydrates are classified into the four category that we are already discussed about it. The first category is your monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides means that mono means single and saccharides means sugar. Okay, so monosaccharides is the single unit of sugar, means that we cannot hydrolyze it. That is your monosaccharide. For example, glucose. Okay, disaccharides have a two units of disaccharides have the two units of the monosaccharides. Okay, we can say that the disaccharides are made up of the two units of the monosaccharides. For example, your sucrose and lactose. Okay, they are made up of the two type of that um, uh, monosaccharide. That is, glucose is made up of, sorry, sucrose is made up of the glucose and fructose. Oligosaccharide consists the 3 to 10 units of the monosaccharides. Okay, and polysaccharides consist of many units of the monosaccharides. For example, your starch and cellulose. Monosaccharides is the simplest form of the carbohydrates, or you can say that it is the smallest unit of the carbohydrate. Smallest unit of carbohydrate or simplest sugar which cannot be hydrolyzed. Which cannot be hydrolyzed means that we cannot break it further. Okay, so this cannot be hydrolyzed, and their classification are based upon the two way. The one is your presence of the carbon atom. It is divided into the three category: trioses, pentoses, and hexoses. Trioses means that the three carbon present, in pentoses 5 carbon is present, and in hexoses 6 carbon is present. Second classification is based upon the presence of the functional group. So if the aldehyde group is present, then it is known as a aldose, and if the ketone group is present, then it is known as a ketosis. So we have to discuss about this aldoses, hexoses sugar, that is your glucose. Glucose come under the category of the hexoses and aldoses, means that 6 carbon are present and free aldehyde group are also present. What is hexoses? Hexoses means that monosaccharides which contain the 6 carbon atom. Okay, here you can see that. This is the formula of the glucose, C6H12O6, which is clearly mentioned that the 6 carbon is present. 6 carbon atom is present. That's why it is known as a 6 carbon atom, and it may exist as a form of aldose or ketose. Okay, I'm talking about the monosaccharide. Monosaccharide can present in the form of aldose and ketose. But if you take the example of the glucose, then glucose is a aldose hexose sugar, means that along with the 6 carbon atom, here aldehyde group is also present. Okay, but if we take the example of second monosaccharide, that is your fructose, in fructose 6 carbon is present, but here the ketone group is present. Ketone group is present. That's why fructose is known as a ketose hexose, and aldose is known as a glucose is known as a aldose hexose. Now come to the next slide. Definition of the aldose sugar. Aldose sugar have a aldehyde group at the terminal of the carbon. Okay, so at the terminal of the carbon, the aldehyde group is present, and this aldehyde group is a free aldehyde group. This aldehyde group is a free aldehyde group. The general formula is CnH2ONON. Okay. For example, glucose, galactose, and mannose. Even they are what? They are the aldose sugar. Okay, here I'm not talking about the fructose because the fructose is the ketose sugar, but glucose, galactose, and mannose is the aldose sugar. The aldehyde group allow them to act as a reducing agent. Why they act as a reducing agent? Because this aldehyde group is a free aldehyde group. When the free aldehyde group is present, then it acts as the reducing agent or known as a reducing sugar. Now, next, structure of the aldose. Aldose contain one aldehyde group and multiple hydroxyl group. Hydroxyl group means that OH. Okay, so when we talk about the structure of the aldose, then it contains one aldehyde group and along with it, multiple hydroxyl group is present. Okay, general formula is CH2OH, CHOH, and CHO. These aldehyde groups react easily, giving aldose their reducing properties. Because this aldehyde group is react easily, why? Because they are free, and when they are free, they can easily um, react with the other um, react with the other reactant and show the reducing property. Now, after the structure of the aldose, what is glucose? So, glucose is the most common type of the monosaccharide which are present into the nature. And this glucose is also considered as the most abundant monosaccharide present in the nature. Okay, this glucose is the most common type of the carbohydrate, most common type of the monosaccharide which we get from the nature. And the chemical formula for the glucose is C6H12O6. C6H12O6. Okay, most abundant and most common. And basically, this glucose provide the energy. Glucose is what? Glucose is nothing, it is a sugar. So this glucose, along with providing the energy to the human being, it also maintain the blood sugar level. Maintain the blood sugar level. This ketose, sorry, this sugar, glucose. This is also known as a dextrose or graph sugar. The glucose is also known as a dextrose and graph sugar, and we get the glucose from fruits, honey, plant sap. And this glucose is the reducing aldose hexose sugar because here a free aldehyde group is present and they have a 6 carbon atom are also present. Discovery of the glucose. So this glucose is first isolated in 1747 by Andres Marggraf. He is a scientist who invented the glucose in 1747, not 1744, 1747 by raisins. Okay, glucose is discovered by the raisins. Glucose is discovered by the raisins. Then after that, during the 19th century, during the 19th century, Emil Fischer established the structure of the glucose. Okay, in 1747, only the glucose is isolate from that raisins. Okay, but during the 19th century, structure of the glucose is established. Fischer also introduced the Fischer projection for the monosaccharides. Now the next is importance of this glucose. This glucose are act as a primary source of the energy for all the living cells. Either they are plant, animals, and human being. For all of them, this glucose is act as a primary source of the energy and central molecule in metabolism. It acts as a central molecule in metabolism, building block for polysaccharide like your starch and cellulose. Involved in the glycolysis, Krebs cycle, and photosynthesis. Glucose is involved in the glycolysis, Krebs cycle, and photosynthesis. Now, the next is your molecule structure of the glucose. So, it can be exist in both type of the structure, that is your open chain form or cyclic form. In open chain form, 6 carbon atom along with an aldehyde group is present, and that aldehyde group is present at carbon number 1. We see that the open chain of the glucose, so we have, first of all, we have a 6 carbon atom. 1, 2, 3, 4, 1, 2, 3, 4, 5, and 6. Then here CH2OH, and here oxygen is attached, here H, then OH, H, H, OH, OH, and OH. It is known as a open chain structure. When we talk about the cyclic form, then it is a hemiacetal ring between carbon number 1 and carbon number 5, and it is represented like this. CH2OH, OH, H, H, OH, and H, and OH. It is a cyclic form. It can also exist in a alpha D glucose and beta D glucose that we already discussed about it. D glucose is the biological active form of the glucose. Now, if we talk about the open chain form of the glucose, so it is a linear structure of CHO, CHOH4, and CH2OH. And this aldehyde group is present at the carbon number 1 and hydroxyl group on the other carbon. It can be oxidized very easily, reason for its reducing properties. That's why this glucose are considered as a reducing sugar, because it can oxidize very easily. Cyclic structure of the glucose. In aqueous solution, glucose mainly exists in cyclic form. If we talk about the aqueous solution, then glucose exists in a cyclic form, that this one. Okay, this one. This one. This aldehyde react with the hydroxyl carbon number 7 and form a hemiacetal formation and form a pyranose ring 6-membered, more stable than the open chain form. Alpha and beta anomers of the glucose. During the ring formation, carbon 1 become the anomeric carbon, and OH on carbon number 1 below the plane is a alpha anomer, and if OH in carbon number 1 is above the plane, suppose that if the hydroxyl group which are present into the carbon number 1 is below the plane, then it is known as a alpha anomer. And if that hydroxyl group which are present into the carbon number 1 is present just above the plane, then it is known as a beta anomer. In solution, glucose is interconverted, that is your muta rotation. D and L form of the glucose. So it is determined by the configuration of the hydroxyl group which are present into the phenylamate carbon. D glucose hydroxyl group present on a right side, L glucose hydroxyl group present on a left side. Natural glucose is D form and they are biologically active. Reducing property of the glucose. So this glucose show the reducing properties because of the presence of this aldehyde group which is present in an open chain, and it can reduce the Fehling reagent, Banach reagent and Tollens reagent and form a red precipitate of the Cu2O or silver mirror. That's why it is known as a reducing sugar, due to the presence of that free aldehyde group in an open chain form. Test for the reducing properties. Fehling test give a blue to brick red precipitate. Banach test give a blue to red copper oxide. And when we do a Tollens test, then it give a silver mirror on a glass. Okay, which confirm the presence of the glucose as a reducing aldose sugar. So for the confirmation of the reducing properties of the glucose, we perform the three tests, either Fehling test, Banach test, and Tollens test. When we perform a Fehling test, then we get the blue to brick red precipitate, which confirm that the glucose as a reducing aldose. Then we perform a Banach test, which give a blue to red copper oxide. Okay. Again, it confirmed that glucose is glucose as a reducing aldose. Then we perform a Tollens test, which give a confirmation of the glucose as a reducing sugar. That is your silver mirror on glass. Now the next is your chemical reaction of glucose. So the first reaction is your oxidation reaction, reduction reaction, fermentation, and esterification. So oxidation test is that test when glucose is converted into the gluconic acid and it is mild. Okay, but when this glucose is converted into the glucaric acid, then it is strong. Okay, here are two conversion is take place when oxidation reaction are occur. The first one is your glucose is converted into either gluconic acid or this glucose is converted into the glucaric acid. Okay, when it is glucose converted into the gluconic acid, then it is a mild reaction, and when this glucose is converted into the glucaric acid, then it is a strong reaction. In reduction reaction, this glucose is converted into the sorbitol. Okay, when the fermentation is occurred, then glucose is converted into the ethanol and produces the carbon dioxide in the presence of yeast. In esterification, glucoside form with the alcohols. Okay, when esterification reaction is occurred, then this glucosides are formed along with that alcohol. Hydrolysis and dehydration reaction. Glucose undergoes the condensation and form a disaccharides maltose. Okay, when glucose is condensate, then disaccharides are formed, that is your maltose. Dehydration by acid form a hydroxymethyl furfural, and this reaction is very important for the food browning reaction. Biological significance of the glucose. It immediates the energy source by the process that is known as the glycolysis. Strong as a glycogen in animals, starch in plants. Oxidized in mitochondria so that it produces the ATP. And it is essential for the brain and red blood cells. Role of glucose in food industry. Okay, this glucose provide the sweetness and energy. It also used in the confectionery industry, bakery and beverages, and involved in a Maillard reaction for color and flavor, and affect the texture and preservation of the food product. Industrial production of glucose. production of hydrolysis of a starch by using acids or enzymes. Okay, enzymes means that amylase and glucolase isomerase, and syrup contain the glucose and maltose, and it is used in the production of the soft drinks, sweets, energy beverages. Medical importance of the glucose. Glucose is used as a administered in IV fuel for quick energy recovery. Okay, this is very common use of the glucose in a medical, that it is administered as a IV fuel, and it give quick energy for the patient and for their recovery. It is also used to treat a hypoglycemia in which the glucose level is low. Component of the oral rehydration solution, that is your ORS, and monitor the blood glucose is a vital in diabetes. Analytical determination of the glucose. So the first method is your reducing test, that Fallin or Banning test, colorimetric, that is your DNSA method, and enzymatic, that is your glucose oxidase test, and this can be used in a food quality control and clinical diagnostic. Muter rotation of glucose. It can change in optical rotation in aqueous solution, and interconversion between alpha and beta form. Equilibrium mixture, 36% of the alpha and 64% of the beta form, and it confirms the cyclic nature of the glucose. Optical activity, glucose show the optical activity. D-glucose rotate of polarized light to right. That's why it is known as a dextrose. Role in metabolic pathway. So central molecule glycolysis produce the energy. Glucogenesis produces the glucose formation. Pentose phosphate pathway, that is the generation of the NADPH, and glycogenesis and glycogenolysis. Glucose and food browning. It participate in the Maillard reaction with the amino acids, responsible for the brown color and aroma in a baked food, and it is very important for the development of the flavor in a food processing. Summary: glucose is an aldose reducing hexose sugar. It can exist in a both form, that is your open chain or cyclic form, and play a vital role in a different process, that is your metabolism, energy production, and food chemistry. And it can be used in a different industry, either it is a food industry, fermentation, and medical industries. So this is all about the introduction of the aldose hexose reducing sugar, that is your glucose. I hope you will understand, but if you have any query related to this, you are freely to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.